My name is Curt Scharfe, and the title of my talk is Development of Multiplex Viral Capture and Sequencing. Viral infections are a leading cause of morbidity and mortality among immunocompromised children and adults. A large number of potentially disease-causing viruses remain uncharacterized, and clinical testing often requires testing for several viruses. Current methods for testing in the clinical laboratory typically rely on single-target nucleic acid amplification tests. These tests employ quantitative, real-time PCR for detection, which is a highly sensitive and specific method and the standard of care. However, qPCR assays are singleplex or low multiplex due to fluorescent reporter molecule availability and difficulties in optimizing primer probe concentrations. This slide shows the workflow for clinical viral molecular testing. It starts with sample collection in the hospital, sample preparation in the clinical lab, viral testing using qPCR, and data review and sign-out. In order to detect multiple viruses from small clinical samples, we have previously developed a multiplex PCR-based targeted next-generation sequencing assay called MV-seq. MV-seq was first developed for the multiplex detection of 20 transplant-relevant viruses, DNA viruses, in 2018. We showed that MV-seq's performance was comparable to that of qPCR for DNA virus detection. In this project, we were interested to repurpose the MV-seq technology for molecular testing of SARS-CoV-2. There were two main challenges. First, MV-seq had so far only been validated for DNA viruses, while SARS-CoV-2 is an RNA virus. Second, due to shortage in RNA extraction reagents earlier this year, we decided to investigate sample processing methods that do not require RNA extraction. This slide shows the MV-seq method adopted for SARS-CoV-2 detection and sequencing. Starting from an inactivated VTM buffer sample as a template, we perform a two-step PCR protocol. It starts with a single-tube, two-step RT-PCR, followed by a second PCR using the virus amplicons as a template to generate a sequence-ready library, which can be readily run on an Illumina sequencer. Notably, the products from the first RT-PCR can be visualized on the Agilent bioanalyzer. The products in the multiplex reaction were designed to have different lengths. The bioanalyzer results can be generated within 2 hours after a sample is received. This slide shows the results from our 4-plex single-tube capture of 3 COVID genome target regions and the human RP gene. The upper figure has the normalized NGS reads for 11 patient samples with different viral loads, ranging from high to low viral loads from left to right. This is based on their qPCR cycle threshold, CT, shown below for each sample. The 4 samples on the right are negative VTM samples and no template controls, respectively. The 2 smaller panels on the bottom show the bioanalyzer image for 1 sample with higher viral load on the left side and lower load on the right. Summary and next steps. We designed 4-plex multiplex PCR for SARS-CoV-2 detection directly from VTM specimens without RNA extraction. We identified all high viral load samples corresponding to qPCR cycle thresholds up to CT32, which approximately corresponds to 500 copies per reaction. We are planning to test additional low viral load samples in the range of CT32 to 35. We are also planning to test assay performance for extracted RNA samples in comparison. We will compare our results to other NGS-based methods currently being evaluated at YCGA, including SWAP-seq and Illumina's COVID-seq. We're planning to develop SOPs, standard operating procedures for robust and reproducible workflows. The assay can be expanded to include other common viruses, for example, influenza A and B, and to enable detection of co-infection with multiple respiratory viruses. We would like to thank these colleagues and collaborators who have helped and supported this project. Please contact me if you have any questions or suggestions. Thank you for your attention.